Good morning, everyone. It's my great pleasure to give you an overview on the technology that I have developed during my PhD. In fact, I'm a cell biologist by training. I did my master's and PhD at the Department of Urology in Medical University of Vienna, in Austria. And I developed a technology that brings the idea of flow cytometry, so quantification of antibody-mediated staining on the single cell level from suspension to tissue section. And this is what tissue cytometry, it's also called tissue image cytometry, is referred to. And today I would like to give you an overview of the applications. A taste, let's say. There are way too many to mention them all. I will also refer to my work here at TRI or at QUT in my role as adjunct professor. And as you may have heard, some of us as a who is TissueGnostics? It's a medtech company certified for ISO 13485, the main quality norm for medical products, with its corporate headquarters in Vienna, in Austria, in this nice building from 1890, very close to the city center. But as already has been said, we have today global structure of offices and subsidiaries. And in fact, I'm setting up, or we have set up last year here, TissueGnostics Australia division. And as many of you have or may have heard, we currently have a demo system here at TRI in level 4 in my Mark Scott's microscopy core facility. As what is called the TissueFAC Spectra, a scanning system for multispectral imaging. And if you would like to know more about it after the presentation, I will not talk too much about the product. Actually only about the applications that have been done. I invite you to contact my staff member here in TissueGnostics Australia division, Sanjit Seth. He's an application specialist. You may anyhow know him because he worked here at TRI for several years as an RA in the group of Jyotsna Batra, level 3. So the instruments, just that you have an idea, are based on a microscope. Can be used for bright field scanning, fluorescence. We also have a configuration with a spinning disc confocal for doing whole slide confocal imaging. There's an inverted setup where you can do live cell imaging and time kinetic assays, or a high throughput configuration with a slide loader for 120 slides. The system we have at level 4 is this one with a liquid crystal tunable filter that allows multispectral imaging of 8 markers simultaneously stained on the same sample. So what is tissue cytometry? I already said it's the idea of flow cytometry brought to the slide, to the tissue section, where by use of image cytometry, we automatically identify individual cells in tissue context and quantify how much staining is associated with it. We can also look on multicellular structures like glands, glomeruli, tumor foci, and then make connections to, for example, tissue-infiltrating leukocytes, which is called spatial immunophenotyping or spatial biology in a sense. We refer to it as contextual tissue cytometry. As it is a high-resolving microscopic system, we can also look into single cells, into subcellular compartments like nuclei, cytoplasm, cytoskeletal elements, mitochondria, or also look for pathogens like viruses, bacteria, or intracellular parasites. Why is quantification important also in microscopy? I mean, we all scientists, I don't need to explain why quantification is necessary in general, but for many years, microscopy has just been used as a means to get nice images. So when I think back at the time of my PhD in the 1990s in Vienna, I had many cases. I was an operator of a Zeiss confocal microscope there. And there were many times researchers, even from other departments, came to me after they had practically finished their manuscript based on data from whatever fax, ELISA, Northern, Southern, Western, whatever was available by the time. And in the end, they came to me and said, Rupert, I need a nice picture. And I said, hold on, microscopy is more than a microscope is more than a lens for a photo camera. It's a means to generate raw data. We just have to extract them from the image, but in a proper way, and that way is image analysis. So the famous proverb that we all know, seeing is believing, shall be challenged and questioned. Shall we really believe everything that we can see? And let me give you a few examples. This is obviously artificial image where we show or where we see individual regions with different gray values. Gray values are important in microscopy because this is what the digital cameras nowadays detect. And especially when we look on fluorescence images, these are monochrome cameras that capture the images, and in staining intensity is referred to as gray value. 256 shades of gray in an 8-bit image, or 65,536 shades of gray in a 16-bit image. But it's always about gray values. Now, let's look on these two regions, A and B here. And what would you say is the difference in gray values between these two areas? For the human eye, it will not be possible to see a difference of, say, 37 shades of gray. But in the scope from 0 to 255, 0 being black, 255 being white, what would you guess which of these 5 answers is correct when you look on A and B? What would you say? Sorry?
Less than 50.
Less than 50, okay. Any other opinion? Answer number 4, so less than, so somewhere between 50 and 100. Okay, so the good news is you are in the global average. Most people think that an answer 3 or 4 is correct. The bad news is you're wrong. Because there is no difference between these two areas. This is not a trick, it's a phenomenon that is called optical illusion. When we compare A with B in this direction, we see a difference. This is apparently darker than this one. And I guarantee you, I can also see the difference, although I know it's not there. If we compare A with B in this direction, we see there is no difference. So this comes from the fact that this region A here is surrounded by brighter areas, while B is surrounded by even darker areas. And by looking at it, our brain, which is a very powerful interpolator, gets active and starts to show us things which actually do not exist. Now, let's assume we would be looking here at cells in a tissue section, stained for whatever, tumor marker, cytokine, whatever you like. And we would see here a cell in a high background environment and conclude it's, say, tumor marker negative, while this cell would be in a low background environment and we would say, oh yeah, that's positive. So we would have one cell negative, one cell positive, and we would be 100% wrong. Whether it's for positive or negative is not the question in this case. The point is it's the same and not the difference that we visually perceive. So the take-home message of this slide is that our perception when we look on structures of interest may not be determined by what we are actually looking at, but by the environment. And this may cause a lot of false data. And also when we look now on a specific microscopic image and immunohistochemical staining of colon with a well-known marker, so the peroxidase DAB staining system, the marker is Ki67, so proliferation marker expressed in the nuclei of cells. Then I have another question for you. When we say, okay, all the cells that we see here are 100%, and how many in percent are brown positive? What would you say if you were to judge this staining? How many cells are positive? How many cells are proliferating in percent? 1, 5, 13, 20, 10. Okay. 1, 5, 10, 13. Okay. Now you are a little bit below the global average, which, as most people believe, there is between 15 to 20. But when we did a survey among 200 experts, researchers, and the range that we received was this 1 to 14, which is, I would say, a substantial difference, but it's also very normal because our perception is influenced, as we saw in the previous case, by many things. It might even depend on the light conditions that are around you when you analyze this image. It may have an effect whether you do it in the morning, before coffee or after coffee, or in the afternoon. So there are many things that influence our perception. And this is the usual thing that we see with visual evaluation, get a range of estimations, and rather than a reproducible value. I also discussed this once with a group of pathologists, and they said, when you ask 10 pathologists, you get 11 opinions. This also points to the problem. In addition, there has been this famous study or survey published published in Nature some 10 or 8 years ago, I believe. Well, there was a survey among 2,500 scientists, and 70% of the researchers indicated that they have problems or fail to reproduce published data. And another 20% said they even have problems in reproducing their own data. So the question was raised, do we have a reproducibility crisis? And there is kind of, and that's, I'm not discussing this in particular, but I'm just showing this as an example why analysis and quantification, also in microscopic images, is essential. And why we have developed this platform for tissue cytometry or tissue image cytometry. If you now analyze exactly that image that we saw earlier with this tissue cytometry system, it will use the nuclei as what we call a master channel to identify the individual cells, and then measure how much blue or brown or whatever color it is are present in each or are associated with each of the nuclei, and show this the same way, from flow cytometry in form of this scatter plot, what people in fact would call it a dot plot, where one dot here represents a single cell in this tissue section. We have on the x-axis now the mean intensity of hematoxylin, and on the y-axis the brown staining, so the Ki67. There is this very important line that we also well know from flow cytometry, the cutoff, that determines the specificity of our staining. And this is very important that we set it properly, because if I set the cutoff here, I will get 0.1% of cells positive. If I set it there, I will get 99% of cells positive. So it's not a matter of taste where I set the cutoff. It's the same procedure that has been established in flow cytometry, which is called the isotype matched negative control, on which you set this cutoff and then apply it to your samples. And this will give you the number of positive cells. And the good thing is you will always have the same number, whether you do the analysis on Monday morning, Wednesday noon, or Friday afternoon, here in Brisbane, in Sydney, in Vienna, in New York. Now, the need for quantification is also the link from me to QUT, or from TissueGnostics to QUT. Together, I'm currently working with Jyotsna Batra in her lab at level 3. And together we have defined a project that we call the Virtual Histopathologist for Precision Cancer Diagnostics. That is an AI-based decision support system for histopathology. And the model disease that we have decided, because obviously Jyotsna is working on prostate, and I, as I may remember, I said I did my PhD at the Department of Urology. So also I have a personal link to prostate cancer research. This is our model. The project consists of several sub-projects. One is that we are working on a multiplexing staining technology. You know, nowadays it's very important that we offer solutions to look at multiple markers. For a phenotypic characterization in context of spatial biology, we need to apply many markers on the same sample to properly characterize cell types and their interactions. And while in flow cytometry or in spatial biology, I see we have a Ruther here, you are used to having thousands of genetic markers on the same sample. With functional analysis, we're still limited to, say, dozens in best case. And these dozens also can only be obtained by serial staining and not by simultaneous staining. With multispectral imaging, we can do maybe 8 to 10 markers simultaneously. If we need to do more, we have to do multiplexing. And the challenge of multiplexing is that in each staining round, you have to use the same set of fluorochromes, but linked with other markers. So you need to make sure that in round 2 and 3, there's nothing remaining from the signal in round 1 or also from the previous round. So we are working here on a linker technology, which allows to reversibly link and remove the fluorochromes from the antibodies, and this should work in a quick manner. One approach, for example, is photobleaching, but this has a lot of disadvantages. Also, we have a collaborator at the Department of Erlangen, at the Department of Dermatology in Erlangen, Germany, who uses the MELK technology, MELK multi-epitope ligand cartography, where they demonstrated up to 200 markers sequentially stained on the same sample, always using FITC and PE, 2 fluorochromes, which can be quite nicely bleached. The second project here within this virtual histopathologist, the second sub-project, refers to integration of genetic data. This means that we have like RNA-scope, algorithms for RNA-scope, but also we are in discussion with the companies like NanoString or 10x Genomics, or the Murphy's technology, that we can import data from these single-cell genomic platforms and combine them with our phenotypic platform. The core of the whole histopathology project is, however, machine learning or deep learning to automatically correlate molecular markers. So this is what is called a chord diagram. The scattergram that we saw before is useful also from flow cytometry, as you know it, when you have a few markers. But then you can see one marker against another marker against the third and the fourth. Right? But when we have dozens or even hundreds of markers, you're lost with scattergrams. Then you would need to go to tens of thousands of scattergrams to find correlations. This is not possible. So we need other means to find correlations. These are the names of the molecular markers that have been stained, and the lines here indicate how the markers are correlated with each other. So the lines, the colors, the thickness, and how the lines go show which marker is correlated with which other marker. And this is done by what we call data mining using machine learning technology. This is already under development, but will be further expanded within this project. And there is also another sub-project, which is, I would call it purely academic. It does not really cannot be sold, but it's still important. Also from a commercial point of view, when we talk about regulatory bodies like US FDA, that we try to better understand what actually happens in a deep neural network, where we define the input and see what comes out, but what happens in the network is a black box. And I want to give you simple example that we experienced a number of years ago. We always had a classical nuclear segmentation algorithm that we use to identify the individual nuclei. In this case, it's a DAPI staining. This worked very well when the cell density was like here. But when the cells become very dense, we ran into problems, because cells were overlapping. And you can see here the recognition. Here it's pretty okay, it's very nice. But in these dense areas, and these dense areas are typical in lymphatic tissue, for example, it was really problematic. So we were up to develop a deep learning based nuclear segmentation algorithm. And as you can see, the recognition accuracy of this DNN is highly superior. I'm not going to tell you the accuracy is 100%, but if we compare these two images, I think it's very clear that the deep learning algorithm is superior to the classical segmentation algorithm. So when we saw these results, we were like, yeah, we did it. Like in the Lego movie, everything is awesome. And then we realized it's actually not. Because one day we came across this sample. It's a very simple staining, actually only one marker in brown, which is expressed or supposed to be expressed in the nucleus. There's a lot of background here. Some of the nuclei are negative, and the client said, how can I analyze this? And we said, okay, with our new nuclear segmentation algorithm, this should be not a big problem. And in fact, it was not. I mean, you can still discuss that there are some wrong segmentation artifacts here. But overall, I think it did it reasonably well. But what came then blew our mind, because the deep learning algorithm found a lot of phantom cells, which are actually outside the tissue. And we were, again, like, what's going on here? And we had a closer look, and this is what I'm referring here to. We defined what we give the algo, and we saw what comes out without really understanding what happens in there. Nowadays, you find many published deep learning algorithms, open source, and you can use them and modify them, but it's always the same story. You define this one, you may change the algo in a way here so that different interconnections appear, but as a human, we hardly understand what really happens. Then we see what comes out. This is the black box I'm referring to. Now, in this case, we actually we analyzed the situation, and we found there are two aspects why we get phantom cells. One is, you may see that there are many dirt particles. And in the pre-processing of this algorithm, we apply a filter to diffuse the image a little bit. And the dirt particles became then diffused and had more or less the size of small nuclei. And the algorithm was trained on also finding small nuclei and nuclei which are stained very thin. But still we thought, okay, why does it find this as nuclei? The next thing we found was that we missed something. The training of this algorithm was done on a data set of H&E stained samples from multiple tissue acquired in multiple magnifications, so that it finds not only cells stained with acquired with 20x, but also with 10x or 40x. But what we missed and what the algorithm never learned is that there can be a part in the image which is empty. Because all the training images that we used showed tissue on all the pixels they consisted of. There was never an outside of the tissue or a hole in a tissue. The whole image was tissue. And so the algo kind of learned, okay, everything is tissue. And the data entity, empty or outside, was unknown. So this is a trap, and I mean, looking back, it's a funny thing, but by the time we were quite shocked, obviously, that this deep learning produces such a garbage. And this is something where I also would like to invite you, if you're interested, to join the project or be as a sample or disease-specific researcher or as an AI-related researcher here at QUT or also UQ or whatever, just contact me either on my company email or on my QUT email address. If you have markers which are not expressed in the nucleus, like the K67 example we saw earlier, but for example, leukocytes or other markers in the cytoplasm on the membrane, it's a different strategy. We still use the nucleus as a master channel and then look what is around each nucleus. It's a so-called ring mask. This can be applied to leukocytes. This is an example from a client at University of Western Australia. They're looking at regulatory T cells, CD4 FOXP3, and we use DAPI as master channel. See here what the system identifies, and then measure for each cell how much yellow, red, and green is associated with it. And this is shown in this type of scatter plots. This would be the CD4 positive population. There is a feature that allows you to track back individual dots or groups of dots in a so-called gate back to the image. So what you see here in red are the cells which are inside the gate. CD4 is the yellow staining, so you will have some which have a very strong CD4 positive, the strong yellow here. This will be a dot up here. Others that are only faintly stained in CD4, they will be closer to the cutoff, but still above the cutoff and therefore referred to as CD4 positive. The rest of cells which have a green contour belong to the CD4 negative population. The same thing we can do with CD8 and ultimately determine the co-expression of CD4 FOXP3 double positive cells. And you read it really like in the FACS plot, the quadrants, the 4 quadrants here, upper left, upper right, lower left, lower right, double positive, double negative, and single positive. This link between virtual slide or image and data space works in both ways. You can also click on any cell in the virtual slide and it will start blinking in the scattergram, so you know exactly where this cell is located. We're looking here on a renal allograft rejection. By the way, in green we have T cells and in red we have monocytes. So you have an immediate link between a cellular phenotype and its location in the tissue. This is forward and backward connection or reverse gating. This system has been applied, and you can look on the TissueGnostics website in the research section. There is a searchable database because we track what our users in more than 60 countries around the world do with this platform. And we have now more than 2,300 papers published from our users. And they're really from basic research in mouse to clinical studies for pharma companies. You can find everything, including virus load on individual cells. In this paper, individual virus particles were counted, or in this publication, they looked actually on the viral load. But I'm showing this to make it clear that with this system, you can also trace minor cell populations. It's not only useful when you have a marker that is expressed on 40% of all cells, but also when you're looking here on, you see 0.06, 1.39, 0.55, 0.21. So also for these minor cell populations in whole slide imaging or images, you can do the quantification. A specific example now for multi-spectral imaging. This is the system in Microscopy, which is currently in Microscopy's facility on level 4. This allows to unmix the overlapping spectra of up to 8 fluorochromes. And when you apply 8 fluorochromes at the same time, you cannot avoid that the emission spectra overlap each other. Yeah? So you cannot just use bandpass filters to detect. You have to apply a mathematical procedure that is called linear or spectral unmixing that allows you to compute how much from the total emission that the system detects using a liquid crystal tunable filter comes from which of the fluorochromes. There's a very prominent reference paper for this technology coming from Harvard University, where they used at the beginning of the pandemic in 2020, they used this system to quantify the immune response in patients who died from COVID-19 in the lung. So to quantify the immune response in the lung by using a 7-color staining. I don't have the time to go into detail, but I just would like to mention this was, to my knowledge, the first, or for sure one of the first papers that indicated that we have a problem with long-term immunity in corona, in the corona pandemic. I mean, nowadays we all know it, but thinking that this was published online in August 2020, so just a few months after the onset of the pandemic, when some countries or some governments in some countries still believe they don't need to do anything, they just wait for herd immunity to come. This was an early indication that that strategy will fail. I would also like to refer to a technology which I find very exciting when we consider increasing the resolution. The spectral unmixing is a way to increase the spectral resolution, to separate spectrally overlapping fluorochromes. But also when you want to increase the lateral resolution in your system to see events which are very close to each other, you can, of course, increase the magnification. You can use, in our case, the spinning disk confocal to do whole slide confocal imaging. You can get a super resolution microscope, which costs maybe $1 million on top of your microscope, or you use other technology, which is called expansion microscopy. This has been developed by the group of Edward Boyden in Boston. And although it's called microscopy, it's actually not a microscopic method, but a chemical method. The idea is that you use a polymer. So you have your tissue section stained with fluorochromes. You put it into this polymer solution, which hardens out. And then you can expand this scaffold. The polymer solution captures the fluorochromes from the tissue, and then you expand it. It's like, I always say, it's like the expansion of the universe, but on your slide. Or you can compare it when you have a balloon and you blow up the balloon. Then the more you blow it up, 2 dots painted on the balloon get further apart. And this is what they are doing here. So by using a standard microscopic equipment, or in, for example, our confocal scanner, then you can combine this with this technology to get into the domain of super resolution. And I want to show you an example. Can we dim the light a little bit? Yeah, this is better. Thank you. This is a prostate, obviously, and we see here the individual glands with cytokeratin, the secretory epithelial cells, and then basal epithelial cells. And this tissue has been expanded 4 times. So this is the minimum. The maximum is 20 times, and scanned with a 40x water immersion lens. So we have a resulting effective magnification of a 160x. We all know that there is actually no 160x lens. Even leading microscope companies like Carl Zeiss, which we are OEM partner of, will end with 100x. But here we get 4 times 40, 160x magnifications. We have 4 markers stained: DAPI, cytokeratin, red, and white. They did not disclose which these markers were. But what is important to understand is that this virtual slide here, it's not the full slide, but the part of the prostate section, way more than 1 field of view, is only zoomed out to 25%. Now we zoom in, and you may see it here. So this is 25%, so this is 50%, and this is 100%. 100% means that now 1 pixel on the monitor or the image relates to 1 pixel on the camera chip. And what you see here is an incredible amount of details. Even the chromatin structure inside the individual nuclei, the cytoplasmic structures here in the secretory epithelial cells, and these stars here, or you may say they look like dirt. It's not dirt. These are individual secretory vesicles. So we really see individual secretory events here into the lumen of this single prosthetic gland. And this combination of confocal, but still standard, whole slide imaging and expansion microscopy is what we have called expansion-based super resolution. And in contrast to the real super resolution, you can apply, it's cost effective, you can apply it to whole tissue sections, not only to just a few fields of view, and to multiple markers. Even you can actually even combine it with multispectral imaging. So bringing all this together, scanning and image cytometry, with Sanjit will be able to show you applications in spatial immunophenotyping and looking like in particular on the tumor microenvironment. This is colorectal cancer, which we received from St. John of God Hospital in Perth, stained for 5 markers. Dark blue is DAPI, light blue is cytokeratin, the tumor area, red CD4 T helper cells, green FOXP3, and white here, shown in white, Ki67. The question this research group in Perth addressed was how many CD4s are present, how many are intratumoral, and how many are in the tumor stroma. And if they are in the tumor stroma, how far are they away? So as shown before, we identified all the nuclei in the entire tissue section. The tumor area shown here in this orange mask, identified based on the cytokeratin staining. And then we can combine these masks and ask all kinds of questions, like exactly how many CD4s are there and where are they. What we get in the end is this type of scattergram, where we have CD4 as an input gate. That means each single dot that you see here represents a CD4 positive T helper cell. All cells get identified, but in this scattergram, only the CD4 positive cells are shown. On the x-axis is the size of the nuclei for each of the cells, and on the y-axis is the distance in micrometer from the next tumor area. Now we can make gates here, because this is a distance. So the further we go up, the further the cells are away. 1 to 25, 25 to 50, 50 to 100 micrometers, and so on. And using this reverse gating feature that I mentioned earlier, we can now see here in red all those CD4 positive T lymphocytes, which are just outside the tumor, but not further away than 25 micrometers. Or here, those which are 25 to 50, 50 to 100, and so on. And if we go back to the scattergram, you see here the quadrant statistic, as it's called, also in flow cytometry, where in total we have 7,234 CD4 positive cells. In the gate 1 to 25 micrometer away from the tumor, we find 2,267 or 31.34% of the cells, and so on. You see this for each distance zone, and we can see there's a clear gradient. So instead of looking at it and saying the infiltration rate is 2+ positive, and most of the cells are close to the tumor, you get a quantification: 7,234, 2,267, 1,593, and so on. This is in percentage, and this value here is the normalized cell density, because the total count, I mean, this is all the same sample, so this is relevant. But when you compare samples, of course, the total count is also determined by the size of your section. 1,000 cells can be a high, 1,000 CD4 cells can be a high infiltration rate when it comes from a small area, or it can represent a low infiltration rate when it comes from a full tissue section. So this will show you the normalizations, number of cells per square millimeter of tissue analyzed. Will show you what is the normalized density of a certain cell population. Then, coming to the last example, here we see, by chance it's again colorectal cancer, but this comes from a client in China with a immune checkpoint staining, multispectral. This is the raw data set. After spectral unmixing, you see very nice spectrally clear picture. These are the individual channels that are combined here in the overlay. And now really see that for each cell in the entire tissue section, you can have this link. Where is this cell in all of my scattergrams? Yeah? And what you can do, similar to flow cytometry again, is where they call it gate arithmetic, that we call them input gates, that you define a gate in one scattergram, which is the input gate in the next scattergram. And then you take, for example, the whole quadrant and define this as input gate in the third scattergram. So what we're seeing here is in the first scattergram is PDL1, actually it's the cell size here on the x-axis, and PDL1 staining intensity on the y-axis. So the gate here in green identifies all PDL1 positive cells in our entire tissue section. And this gate is the input gate for the second scattergram, where we, however, look on CD4, the x-axis, and PD1, so this is PDL1, this is PD1 on the y-axis. All events here, because they come only from this gate, we know are PDL1 positive. With our two cutoffs, this is the lower left quadrant, the double negative. We define this as input gate in the third scattergram, where we see CD8 versus CD68. And if we then again look on the double negative here and make this backward connection or reverse gating, then we see here with red contours, where are the cells that are PDL1 positive, CD4 negative, PD1 negative, CD8 negative, CD68 negative. And you can obviously do this for each combination or each quadrant. That means you get an incredibly detailed readout. Of course, all the data, the raw data that are behind these scattergrams can also be exported in a kind of list mode and then analyzed in one of the big statistic applications like SPSS and do further analysis. We also support the what is it called? The fax software. FCS Express. Thank you. From De Novo Software in California. They have a version FCS Express image cytometry where you can import this data set and then make all these like T-SNP plots and that you may know from flow cytometry. So coming to conclusion, I hope I could show you that with this tissue image cytometry, you can really do a quantitative analysis on the slide of your microscopic images. And this can help us to move from image to data and not just use microscopic images as nice pictures in our publications. And thereby increase the observer independence and reproducibility. And again, I really invite you to contact either me or Sanchit directly if you would like to have a look at the system or at the software. The software, by the way, is also available standalone, and we can import images from most of the third-party slide scanners like Aperio, 3D Histec, Olympus, Hamamatsu, whatever they are called, or even just images that you acquired 10 years ago on a confocal. You can just read the JPEG, TIFF, bitmap image, and analyze them. I would like to acknowledge our academic collaborators from, I didn't talk about this, but we had in total 6 EU-funded Marie Curie innovative training networks, or doctoral networks as they are now called, on different topics like on the calcium sensing receptor, the anaplastic lymphoma kinase, cell migration, big data analysis for clinical benefit. This was headed by Medical University of Vienna, this by University of Cambridge in UK, this by Technical University of Aachen in Germany, this by Trinity College in Dublin, where TissueGnostics always was an industry partner and provided solutions on highest level in terms of image cytometry or tissue image cytometry. And in particular, I would like to thank Jyotsna and her team here at TRI, with whom we have set up this virtual histopathology project, and also some partners overseas. With this, I'm at the end. Thank you very much for joining this morning session, and I'm very happy to take questions or make appointments with Sanjay.